mainly we were studying about the databases. Okay, so we were studying about primary databases. We are going to, I hope you can see NCBI website. Is it? Can see NCBI?
Yes, sir.
Yes, yes, yes. So we will be studying first. There are some popular databases NCBI has. We have gone through PubMed, Bookshelf, and PubMed Central. We have already studied, and now we are going to study the database where nucleated sequence has been stored. These are all databases. You can go one by one and check. I'm just going through the important databases only. The database where the nucleated sequences are being stored are called GenBank. You can see I have just shown some trypsin or something. So nucleated, you can select the nucleated from drop down menu, or else if you can click here also, it will go directly to the nucleated database. So you don't need to select anything now. So it will directly go to the nucleated database. You can see nucleated database is a collection of sequences from several sources including GenBank, RefSeq, um, RefSeq, TPA, PDB, genome, gene transcript sequence data provide foundation to the biomedical research and discovery. So we can go to we can find a gene of trypsin, trypsin alpha, trypsin chain 1. We can see in GenBank. Actually, you can see Arabidopsis thaliana chromosome 1 sequence. It is very big sequence. So we need a complete coding region, coding sequence CDS. We have already taken human CDS of this trypsin. We have already taken yesterday. So when we take this, we can see it comes in a format called GenBank format. This GenBank format is a we have said that different databases store data in specific formats. So that makes the specific order or specific formats the data has been stored. This GenBank format is, I have said that database data can be stored in 3 forms as flat files, relational database, and object oriented database. This is an example for flat file. This GenBank format. We can see locus here. Usually GenBank format can be divided into 3 areas. The from locus to journal, this part, it can be called as header, where it define what is that sequence is about, and where it got published, who is the author. All these things we can get it from the header region. So and then comes the gene annotation, some sort of a small sort of annotation we can see here. Gene annotation can see in this part. As it is a primary database, there is not much annotation we can see, but what is the source, which organism is the source, and where we got that, where we isolated that bacteria, or like that source data we can see, and gene data we can see, and where the reading frame starts. All these things, gene annotation can be seen. And the third part is the DNA sequence, which start from a heading called origin, and which ends with this two I will just use my presentation. So we can see this is the one. Usually it starts from a header contain locus contain a identifier number, then followed by the length of the sequence, and whether it is DNA, RNA, or RNA, etc., protein, etc., is being distinguished here, or whether the DNA is linear or circular, it is distinguished here, and the date in which it is submitted, and whether it is belongs to bacteria, whether it is belongs to human, BCT, it is denoted to bacteria. So that identifying letters are also there. Then the name of the sequence is present, then accession number is present. You can see the accession number when we submit one sequence, it can if there any error is there, we can resubmit that sequence. So it will be, I already said that it will be version 2. So accession number you can see U25150. Here version is U25150.1 is a version 1. If it is we resubmit it, is called it will be called version 2, version 3, like that. If any keywords are there, it will be seen here. And where this particular gene will isolated the bacteria name or organism name, and the organism classification we can see the classification bacteria, proteobacteria classification we can see, and how many bases are present in the organism, authors who have submitted, and how they have submitted, and whether whether this sequence has been published in research papers or not. All these things data are seen in the header region. Then coming to the features region or gene annotation region, in which we can see the source of the organism, where it is isolated. Genomic DNA is the source, and whether it is isolated from soil, whether it is isolated from water, all can be included here. All this data we are giving it during the submission. It is nothing, they are not generating any. This is a raw data database, that is a primary database. So nothing, very small things has been generated on the database. That is, this taxonomic classification is usually generated by them only. Most of the things we are entering. I will show you as how to submit the sequence. And gene, where the gene start, we know that 1 to 359 is the sequence, and from the gene usually start from the 79 to 351. That is the coding sequence. It start from 79 to 351. So the 1 to 78 is a non-coding region, and 352 to 359 is a non-coding region. So what are the peculiarities of the gene? Some sort of annotation we can see, some sort of annotation, and the translation of that particular sequence into protein can also be seen in the second part of the GenBank format. Third part start with the heading origin, and we can see the nucleotide sequence in the small letter, not in a FASTA format. I think you know we will come to that. FASTA format will be having the capital letter, and the numbering is not there, line numbering will not be there. So it is not arranged like this, it is arranged in a different manner. So it will end as the sequence ends, there is 2 slashes are also there. This is a GenBank format, somewhat similar format. The GenPept or DNA protein sequence database also follows a somewhat similar pattern. So we have only 21 people. So like that. Is it anyway interesting for you? Because people are decreasing.
Yes, sir.
Can you follow that is because today is Thursday only, but people are decreasing. Okay, anyway we need to proceed. So we have that this trypsin. We can see the same header sequence. Locus is there, definition, that is the heading is there, accession number is there. Here also the version 1 is there, and keywords, usually we give the keywords, it is there, and source it is indicated, and the classification of humans is there, eukarya to homo, and reference where it is published we can see, and the title of the sequence we can see, and if it is published it will be journal, sorry, the authors are there. I said it's publication, sorry. So authors are there, and if it is published, the journal name, everything will be there, and the sequence is of 1 to 1927 bases, and the author's name is there, and title, direct submission, and submitted the institution name, all these things, and comments if we have submitted, it will also come. Then the annotation part will come. That is Homo sapiens. mRNA is the one which we have submitted here, and the taxon, clone, tissue type, from testis only this particular particular trypsin is been isolated, and clone library, that is TST14, cloning vector. This by cloning only this gene was sequenced. So we will see how we can clone a particular gene, how we can design a primer to amplify a particular gene and then further cloning it. So cloning is done using this vector only, and various other parameters. CDS also start CDS or gene. We have seen gene. CDS and gene is somewhat same only. So CDS is start from the 360 to 1780. Rest of the sequence, flanking sequence are not coding region. So its translation, when this genome is translated by computer, we get this protein sequence. This protein sequence we can use it for modeling, etc. Modeling to get the protein model. So we can see the sequence which start with the origin and ends with this these two slash. So this is the GenBank format. It is a very database have its own formats. So apart from GenBank format, there is a most simple format. We can click this FASTA here. The most simple format and the most bioinformatics tools accept this format. That is called FASTA format. Anything GenBank remaining? No, I think. So another important format is the FASTA format. It's the most accepted format by bioinformaticians. But not most of the bioinformatics softwares accept this format. That is a FASTA format. It will start with the gradient symbol, and then followed by accession number, then followed by the title of the paper, and title of the sequence. Title of the sequence which contain whether it is DNA, whether it is RNA, whether it is complete CDS is there, like that. Every details of the particular sequence will be there. From next slide where the sequence will start. So this is how the FASTA format is about. So FASTA is one of the simplest and most popular sequence format, because it contains plain sequence only, nothing annotation or nothing is there, just plain sequence information only, readable by many bioinformatics analysis program. Most of the bioinformatics programs can read this, even it is a PDB, even it is translated ensemble, even it is any database. Most of the homology modeling databases, all of the databases accept the FASTA format. It has single definition line. You have seen the definition line. I will show once again. In another, this GI value is earlier used. GI value was also there in the NCBI, but nowadays they have used only accession number. So genome identification value, nowadays it is not used. So in some sequence it will be there, old sequence it will be, this GA value will be there in the NCBI also. So the single definition line will be there, begins with a right angled bracket, this bracket it will start. I already said it, just reading. Followed by the sequence name, it contains the features of the sequence. Sometimes extra information such as GI number or comments can be given, separated by the this symbol. Extra information considered as optional and ignored the sequence analysis program. Usually we have this first line with all information, but during the analysis every software ignores this line and take the sequence only. It's protein sequence only, this, not DNA sequence. And the plain sequence in the standard one letter symbol, so the sequence will start in the second line. First line will be the title line, and second line the sequence will begin. Each line line of sequence data limited to 60 to 80 characters. So 60 to 80 characters we can see here. One line contains 60 to 80 characters of nucleotides or amino acid. 60 to 80 characters can be seen here. The drawback of this format is called most of the annotation will be lost. There is no annotation at all. Even GenBank format, there is some amount of annotation we can see, but here there is nothing annotation, just the sequence and just a title, that's all. That is the disadvantage of a FASTA format. I have said about the curation annotation. So that is about the FASTA format. So we will continue. So this is about GenBank. GenBank, we can see the GenBank format as well as FASTA format. For every analysis we need to come here and take the sequences. So every analysis mostly what we do today, we need to take the sequences from here and then do the analysis. So this is about the GenBank. So then we will go back to the NCBI. There are more tools, more databases we need to study. Another database is genome databases, which contain genome information. You can see the genome information here. You can see the genome of Homo sapiens. You can see the human genome. Every details of human genome you can see it here. So it will take some time. So these are the some of the most widely used databases. You can see the genome of humans. This is not the one. Yes, eukaryotic rough sequences of different organisms. You can see here we can see we can select human, so that we can see the latest assembly. The human genome, lot of assembly or versions can be obtained. This is the latest assembly, CH38P14, that is the latest assembly which is seen in NCBI RoughSeq. RoughSeq is the database in which reference genomes has been stored. So we can just click this latest assembly of human genome, so that we can see the human genome assembly, and its submitted genomic assembly and taxon and assembly type, haploid with loci and submitter genome reference consortium. 2022 this genome data has been submitted, and their size 3.1 gigabase is the size, and all these details about how many chromosomes, 24 chromosomes, 23 chromosomes, as well as mitochondrial genome has been sequenced, that is 24 chromosome, and one organelle chromosomes has been sequenced, and various scaffold, various details about the genome has been indicated, and you can see the quality analysis of the genome, and single copy genome, 95%, 98% is a single copy, duplicated amount is only 0.7%, so it is highly good quality the assembly we have, and we have the details of each chromosome, 24 chromosomes, and we can go to each chromosomes and see its peculiarities, size we can see, GC content we can see, and localized count actions we can do different graphical view we can go to. So you can go to chromosome 1 in GenBank, we can get the sequence of this chromosome 1. So this is a reference sequence. If we are sequencing you can see still everyone is n like that. So still the sequence is not complete. Many of the regions, then it's just added nucleotide N, because we need to confirm that region, because just they are assuming any nucleotide, arranging any nucleotide in some regions. So that's why we can say our genome is not fully complete. Many regions are still not filled. Many regions are still remaining N like that. So we are in completing or getting the full nucleotide sequence of our genome. So you can see the different genome of chromosome 1. If you have any chromosomal abnormalities or anything, you can associate with chromosome 1. You can sequence your chromosome 1 and we can compare with this reference genome, so that we will get a knowledge about what all changes in your genome has been, and why you got that particular disease or like that things. So this is about genome database. So you will get the genome sequence of each chromosomes directly from the genome database. You can download. It's a big data. If you go to it is very big data. We need to scroll down and down to get that data. So you can go to genome and download the genome data from the genome database. So this is one of the most widely used database. Next is SNP database. SNPs are associated with many diseases. So SNP database mainly contain the single nucleotide polymorphisms associated with many diseases, etc. So SNPs contain human single nucleotide variation, microsatellite, small scale insertions, deletions. All these can be seen in the SNP databases. SNP databases, we can search a gene name or if you know the SNP name, you can directly insert here and search. I will just search one gene name, whether we will get or not. LCT lactase gene is there. So yes, lactase gene is associated with many SNPs. We can, when we search itself, it will go, a drop down need to come, but it is not coming. LCT is a lactase gene. If you just search LCT, this LCT gene, usually LCT is a lactase gene involved in lactose metabolism. This lactase gene usually contain a lot of SNPs. So single nucleotide variation, it is called single nucleotide variation or single SNPs. So this SNP database is the accession number or the identification number is usually start from RS. So RS748 like that it will start, and they are all SNPs associated with this LCT gene has been listed when we just search that particular gene. If you have this RS, you can directly search, so that you will get that RS only. You can just take this. If you have this RS number, I will copy this. If you have this particular SNP and we need to find out the features of this SNP, we can directly enter that RS value. So it will take directly to the this RS only. This is a SNP or single nucleotide changes present in the lactase gene. We can click here, so we can know when we are studying the diseases which is associated with the SNPs. We need to know what all changes, what all SNPs can be associated with that particular disease. So at that time, this SNP database has a great role, because these NCBI database contain most of the SNPs related to many of the genes. So while studying the SNPs associated with a particular gene, we can find out um, uh, we can use this polymorphisms as a marker for detecting that gene, and we can use this polymorphisms, or we can do we can correct these polymorphisms so that a particular disease can be prevented by using gene therapy or some gene therapy techniques, etc. So these SNP database contain lot of SNPs which is associated with various diseases. I think you know SNPs. SNP means single nucleotide polymorphism. Each humans are we two are differ each other by less than 1%. That less than 1% include the changes in the genome, which include the SNPs, microsatellites, all these small changes we can see in the between our genome. So the changes in these SNPs can lead to can result in you are getting one disease and I am not getting one disease. Because of these changes, these single nucleotide changes can lead to a person susceptible to more to a disease. A person is protected from a disease. So we can see the SNP databases where SNPs are being listed. So we can see the Homo sapiens organism, Homo sapiens. SNPs are identified by RS, by identifying number starting from RS, and where this SNP lies, it lies in the chromosome 2 in this position 1, 3, 5, 8, this base pair position. And this according to this version only. Maybe next version of genome sequence come, maybe this number may change, because we may get extra some nucleotide, and this number may change. So according to this version or this assembly, we are getting in this SNP in this base pair. This SNP is look like this. C will be changed to T. Usually, mostly we can see C in most of the people, most of the humans. We can see C as more compared to that of T. So C is high compared to that of the T. So single nucleotide variation. So its frequency, we can see the C is high compared to that of the T. So so intron variant. So it's a intron variant. This variation is seen in the introns only. So like that we can see. So where in different ethnic groups, how this it's the frequency of this particular allele, how we can see. Europeans see T is most widely seen than C. We can see C is less and T is high. We can see C is less and C is less. Sorry. Total globally, C is greater. So is globally total, when we take the frequency of C allele is highly compared to that of the T. We can, when we see Europeans, we can see T is predominates, while when we see in Africans, we can see C predominates than T. So like that we can see different in African others. We can see the C predominates in the particular region than T. Like that we can see in different people. In Asians, South Asians, we can see C predominates, T is less seen in the human genomes. So like that we can see in the these are the studies of 1000 genomes. As I said, 1069 genomes has been sequenced from different ethnic groups. From these studies only we came to know about the frequency of the particular C and T in this variation, this SNP variation. So we can see the graphical representation of this RS. You can see C and T variation. This C is changed into T. This C is changed into T. That is what C and T is being. C is most widely seen, but T is less seen. So C is changed to T in this position. You can see this position in this chromosome. You can see this position. This position 13550, you can see that position number here, where is here. So this position in that chromosome 2, we can see the change in C to T. So the same way we can see a lot of SNPs. A lot of SNPs are there nearby, you can see, but we are just focusing on this SNP only. This RS value all indicates different SNPs. You can see in this graphical representation. This is all about SNP database. SNP database mainly say about the SNPs, and SNPs associated with the different genes. If we find out the SNPs associated with the different genes, we can it can help in understanding the role of SNPs in particular disease, etc. SNPs and gene databases, which give the ideas of different genes. We can see gene databases integrated information from wide range of species. Record may include nomenclature, reference sequences, maps, pathways, phenotypes, genotype, phenotype of the particular gene. We can search a particular gene here, so that the features of that particular gene can be seen here. Which gene I need to, I will go with hexokinase. We can change the gene in between. So here Homo sapien hexokinase 1 we can see. So different features of the genes we can see in the gene database. So a lot of hexokinase gene is present, and in Homo sapiens it is present in the chromosome 10, and so we can just click the particular gene, so that the various features of the particular gene can be visualized during by this page. So official symbol, official name, so kinase primary source, this gene has been isolated. If we click, we will see where this gene has been isolated, and gene type, it is whether it is protein coding or non-coding. Rough seq, it is whether it is reviewed or non-reviewed, whether it is reviewed by humans or whether it is non-reviewed. If it is reviewed only, so it is accurate. So just like the classification is there, organism is there, and summary, what is the role of this is somewhat a annotated database like things, this gene. So expression, where it is expressed, you can see whether it is expressed in heart, it is expressed in heart, esophagus, other tissues, and its orthologs, its similar orthologs can be seen in mouse, and then these are the assemblies, different assemblies where we can see this particular gene, and these are the map of the graphical representation of the particular gene, and this indicates its properties of the particular gene. It can be seen RNA-seq exon coverage. We can see wherever the exons are there, wherever the introns are there. So here the introns are there. These introns and exons can be differentiated here. You can see here intron exon differentiation. We can see here. So and the expression of the particular which protein we have taken. The hexokinase where is expressed, you can see it is expressed more in brain, esophagus, heart, and testis, thyroid, compared to other tissues. So related informations we can go and get its phenotypes, so disease is associated with this impairment of this particular gene hexokinase. You can see its phenotypes, its variation in the particular genes, its interaction with the HIV-1 interactions. So lot of is a annotated database contain lot of links to different sources. So its interaction with the different molecules we can see here. So its general gene information, what's function, it has a lot of function, fructokinase, activity, glucokinase, activity, lot of function, and involved in which processes, and it's a component of where it is present in cytosol, it is present in membrane, present in mitochondria, all these things we can, general protein information we can get from here. A lot of information are there. RF sequence we can get it from here. Hexakinase sequence we can get it from here. So a lot of related information. It's mRNA. So a lot of related information related to the particular gene can be obtained by the gene database. You can when we are studying a lot on a particular gene, better to come to the gene database and check that gene database. So next is next we can go to protein. Protein is somewhat same as that of the as a GenPept. It is called GenPept. So it is somewhat same as that of the genome database. You can just search trypsin. So you can see the protein sequence and it is shown in the GenPept format. I am not explaining anything here. And just like GenBank format, it is we need to check a protein and just taking any protein, a serine protease 1 pre-protein, and pre-protein I am just taking to show you how the protein look like. You can check with another protein. So there will be the same, somewhat same somewhat same way of arrangement. We can see header will be there, and so references and annotation. We can see the comments and annotation feature file is there. So everything is the same only. It is but the difference is that it is a protein sequence only, it is not a DNA sequence. You can see it is GenPept format. This is called GenPept format. So everything you can see it from here. So I am not taking much time. And then last most widely used databases in NCBI is the PubChem. Usually when we drugs, when we are designing drugs etc., we need usually drugs, we design drugs, one of the mainly how we design drug is that we use a small molecule or chemical molecule as a drug. What mostly this drug do is that it will inhibit a protein which is involved in the some important functions of our body, or in the case of corona we have the spike protein. So if we design a chemical molecule which will bind to the spike protein, so that the spike protein can't bind to the humans, so that the corona can't infect the humans. That was the hypothesis which many made, and I also published one paper on that. So spike protein. So these usually the drug molecules, where we can get these drug molecules. So we can get the drug molecules, chemical molecules from a website called from a database called PubChem. We can get we can just search paracetamol. It's one of the most widely used drug compound. Now to decrease the temperature, you can see the paracetamol. You can go to paracetamol, and we will see when we are doing the drug designing. We can get the properties of the particular drug and its various properties, and its 3D structure, its 2D structure, 3D structure, all we can get. Usually we can download the all structures, 3D structure from here, different formats, which can be used for drug designing etc. So mainly the small chemical molecules which can be used as drugs, which could be used as drugs, we can get from the database called PubChem. So these are the database which I am going to explain from NCBI. Most of the some of them are contain lot of annotation. This nucleotide database, that is a GenBank, and the protein database GenPept are purely primary database. So we have already completed the primary database. So we need to submit this data. Okay, how we can submit the data to the GenBank? Usually trypsin, I will just search anything trypsin, or chymotrypsin, another protein. Okay, how we can submit the data to NCBI, that is what we are going to. It's a Homo sapiens chymotrypsin RNA. So that now we are going to study how we can submit the data. Usually after DNA sequencing, we usually get from the sequencing machine this type of FASTA format. So these FASTA format we are now going to submit to NCBI, so that our sequence will also be displayed in the NCBI. So my sequence also I think will be there in the NCBI. I will just search. So we can submit a sequence, no item found. So my sequence was there, but I need to find it out. Anyway, I will find it out. BTCZ10 is one of my bacteria. Maybe I can find out anyway. So as I am trying to say, you can after DNA sequencing, you can submit. Yes, you can see this is my sequence only, one of my bacteria. So you can see my name here. So this is the others. So like this we can see. So this is the data. These all data I have given during submission. Nothing, they have very less data they have added. These all data I have given during submission. They have made it into finally into this GenBank format. Many of you know it already, but I will but it is my job to show you how to submit the sequence to a database. So I will take my own sequence. So usually after the genome sequence, you will get the sequence in the FASTA format. So think it is a FASTA format. So we need a WordPad. So we need WordPad for the submission. WordPad. I think WordPad will work. Notepad is best. Not WordPad. Notepad will work. Not WordPad. Notepad will. So I have just taken the Notepad and just paste this sequence. It is not pasting or anything. Your sequence, if you get from the from the sequencer machine, you just take it from the result mail or somewhere and paste it in a notepad, and you can just add the greater than symbol, and you can just add the details also. Okay, so accession number you don't need to add, they will add. So you can add the details also, and from the second line you can start the sequence and just save this. So I will save as the desktop as a noble one. So I just save as the notepad. Okay. So this is the thing which we first want to do for submitting to GenBank. Then go to Google and search NCBI nucleotide submission. Then you will be directed to a submission portal of NCBI. So all the submission, if it is I am going to submit a 16S rDNA sequence, all the sequence submission is somewhat similar in ways. Very less changes we can see in the sequence submission protocol. So mostly all are similar in their submission. You can see this is submission portal, which where we can submit the particular sequence. You can see GenBank is the largest nucleotide archive containing the sequence of all branch of life. You can, if you want to submit the sequence or assembled sequence of SARS-CoV, influenza viruses, as well as eukaryotic nuclear mRNA sequences, you can directly submit by clicking here. Submit genomic DNA or organelle DNA, plasmids, viruses, phages, the genomic DNA sequence you can submit clicking here. And assembled eukaryotic and prokaryotic genome can you submit to this here. So most of the submissions follow the same. The unassembled high throughput sequencing reads can be submitted through here. So different ways we can submit. So we are just submitting and seeing about a 16S RNA. How we can submit a 16S RNA sequence. So any sequence follows somewhat same way of submission pattern. Every sequence follows same pattern of submission pattern. Before submission, you should log in. You should create a login ID in NCBI and you should log in. First I am just login. Trying to log in, I don't know, I have not done this for a long time. Anyway, I will log in through Google account. I'll try that first, otherwise I will log in by NCBI. So first you log in to the NCBI. That is first thing you need to do. So we are submitting. I am just taking this sequence, but don't submit it. Don't you can study by just submitting until last step, but don't submit it because NCBI won't allow you to submit. So I am already logged into this now. Otherwise, you can create a login ID and log into the NCBI. So I am already logged in here. So then I will click 16S rDNA. 16S rDNA to the GenBank I am going to submit. So unassembled reads of high throughput sequencing with the high throughput NGS sequences etc. you can submit to SRA, or else you can submit to GenBank. Usually I have submitted to GenBank, so I am just showing that. So GenBank submission portal, then it will be directed to GenBank submission portal. So you need to fill all these things so that you can proceed. So take care while filling all these things. All these things will be coming on your GenBank format. So take care while filling this. So the submission tool for the specific data types indicate below. So you can see the submission tool for SARS-CoV, prokaryotic rRNA, eukaryotic rRNA, eukaryotic organelle RNA, meta7, eukaryotic nuclear mRNA. All these things we can see. The data, this is a submission tool for the submission of this much things only, not genome or anything. This much things only can be submitted here. You can just click next. So prepare the following, all these things you need to keep in your hand. Next, so sequence should be in the FASTA format, should also be in this notepad, and you can submit more than one sequence also. After one sequence you can again paste the second sequence. More than one sequence also you can submit, and you can see the source from where you identify, whether you identify from the water, whether you identify from the soil, all these things you can give. And then the processing of the particular gene can also be also be indicated. So all these things can be indicated while you are submitting. So all these are the instructions you should follow, or these are the data you should be take in hand before submission. So when you are ready with this data within your hand, we are going to click the submit. Actually, this NCBI submission portal is called a Bankit. So earlierly called Bankit, now very when we submit the genes etc. only it is Bankit is showing. NCBI keep changing the names, and we can see that names we can't. You can see Bankit here. All other submission you can if you go for submission of genes etc. Eukaryotic genes etc. We can go for Bankit and other tools. So here it is just restricted to ribosomal RNA, meta7 COX1 and eukaryotic nuclear mRNA, and this virus DNA, virus RNA. So for submission, so I have during my teaching a lot of unfinished work is pending. So I have submitted one last 2022. So new submission. So this also will come pending. Click the new submission. You have at least one unfinished submission. Are you sure to create? So I have a lot of unfinished because I am teaching this for the students for a long time. So I have a lot of unfinished submission, but I am starting again because I want to teach you people also. I will not complete the submission. So each I think you people have submitted paper in research journals when they will ask each step to enter different things. Just like that, here also we need to enter different things until it reach the review and submit. In the review and submit region, you can see all the data entered in the GenBank format. That is what we need to keep in mind. What do your sequence contain? Our sequence usually contain the ribosomal RNA. It's prokaryotic or eukaryotic. Our sequences contain prokaryotic ribosomal RNA. Which ribosomal RNA? 16S RNA. So we can give a submission title. Submission title can be 16S. Any submission title you can give. rDNA submission. So just I am giving a title. So we will be going to the, if any error occur, they will not allow you to continue. Okay. Next is the submitter name. So you can give your name, your organization name, and it's already pre-filled, and your name, your contact details, your email ID, your name, and if any others are extra is there, you can create a group and add more others to that, and we can continue this. So the GenBank submission, they have given a temporary ID they have given now. So we have whether you have used any next gen sequencing technology, or I have only used Sanger sequencing technology. So just select Sanger sequencing technology. These are our next gen sequencing technology. We will be studying on 454, Illumina, Ion Torrent, and Solid. These will be studying. Rest of the things you can. If you study that, it's easy for you to study rest of the things. So whether the sequence is unassembled or assembled, we have unassembled only. We are not assembled or anything. So continue. So then we need to what they are asking is that whether we should release a submission on public as it upload. We are uploading. Okay, after uploading, I think by 2-3 days it can be if the NCBI is satisfied with your sequence. If any, they will go through a quality control process. If if your sequence can pass that quality control, it will be directly released. Or else if you are working on that particular bacteria or something, you don't need to show to anybody because a lot of competitors are there. You want to show that bacteria only after publication, you can select this. Otherwise, when you need to then the release date, a tentative date you can give when you need to release. Otherwise, you can release after processing. You can just tell. They will give a check and then they will release. Did you check the low quality? You can check the low quality sequence in the FASTA sequence. If there is any N like that, you can trim using software such as Biodit, etc. You can, if you check, you can tell yes. If you don't check, you can tell no. So cultured or uncultured, whether it is cultured, this gene from this sequence is from cultured bacteria or from metagenomics uncultured bacteria. We have discussed yesterday. So it is from uncultured bacteria only. So we have here to upload the particular file. So where we saved, it's in desktop. We are going to upload that particular file. Where is our file? Noble one. Is the notepad file. So I have uploaded that. Okay, you can see here, and we are continuing. What is happening? They will ask any any problems, yes, they will check the sequence. Okay, then they will, what they are going to tell, they will tell what you are attempting to submit a sequence already have accession, we have just copied from here. So they will find out that that sequence is already in NCBI, they have that accession number KF, you can see the accession number here. So they are giving the warning, but we are just continuing, we will give the warning, we are just continuing now to see the whole submission process. So then they will say multiple sequences can be submitted in one file, one after other, but we are not giving word to that also. We are continuing. Oh, it's not going. Yes, we are continuing that. So then when the next page contains source modifier. Next page contain source modifier. Source modifier is the next file where we can add the organism. I am just filling one organism, E. coli, it's not E. coli, some other organism. So I am just filling E. coli, I can select E. coli from here, drop down, coli. I am just selecting what all organism it is, you can select, give it, and just this is pseudomonas only. Yes, give it. Strain, what strain name you can give, ABC, ACD, ACS2, strain name which you already given, and collection date if you have, you can just give that date, just like 15 April 2023, you can give the collection date. Country we can give India, can give India as country, and note we can add more things, so that it will be coming in the, we can add the latitude, longitude, we can add culture collection identifier if there, you can add that, and forward and reverse primal sequence you can add. Lot of things you can add here according to your wish, can add a lot of the things. So we will be moving from here. Source modifier, if any error they will say, if any error in our fielding they will say. So then we need to give, we have given the institution name, now we need to give the author name. So my name I am just giving. So like this the author name will come. If you need to add another author, you can give that also. So I am giving my name only, and publication status, it is unpublished, in press or published, you can select, it is unpublished only. You can give a reference title, a tentative title which you are going to give your bacteria. I am just saying melanin producing bacteria, this bacteria was melanin producing bacteria. So I will be publishing this bacteria under the heading melanin producing bacteria. Reference author, any the paper author will be differ from that of the sequence author, we can say the same as the sequence author. Continue, we can continue it, so that it will reach the references and it will reach the last step. So when we reach the last step, you can see in this side, you can see the whole data which we have given is converted to, we can submit now, but I am not submitting anything. It is converted to the GenBank format. Accession number they will give after they accept your paper. So all the things which you have added, you can see the feature file, you can see the Escherichia coli you have given, genomic DNA you have given, and strain number you have given, ACS2, all these things you have given only. Country, collection date, all will come in the feature, that is annotation area. They find out the rRNA, they characterize it as rRNA, not gene, CDS. So they characterize as rRNA. Some things are added from their part, but most of the things are added from our part only. You can see how the data which we have given is converted to this GenBank record. They will analyze our sequence and finally accept our sequence. So this is all about the submission of sequence to the GenBank or GenPept. Is submission of any sequence. The genome sequence also follows the same pattern only.